Next up, we have Brandie Morris. She is in the Neuroscience program and works in the lab of Dr. Piera Passanelli. The title of her talk today is Tidying Up Toxic Proteins in ALS.
Imagine you're waiting at a crosswalk. You begin crossing the street. When you stumble, you would probably attribute it to clumsiness and ignore it, but then it happens again and again. With time, you become weak and lose the ability to move your limbs. As your muscles begin to waste, you have trouble swallowing and can't eat without help. Eventually, you can't breathe without a machine. All the while, you're aware of what's happening to your body, but you can't do anything to stop it. Now imagine that the time between that first stumble and death can be as little as 2 years. This is what happens in ALS. ALS, or amyotrophic lateral sclerosis, is commonly called Lou Gehrig's disease. You may know it as the ice bucket challenge disease. What you may not know is that 15 Americans are diagnosed with ALS every day. One of these people was my aunt, who passed away in 2010. Her first stumble happened walking the streets of New York, and she, like anyone else, just thought it was clumsiness. ALS develops when the brain cells known as motor neurons communicating between your brain and your muscles die. I study one of the more common mutations that causes ALS. In this particular mutation, the protein-making factories of the cells go rogue and produce these unexpected proteins. Now, it's unclear if these proteins are the cause or simply passengers in the disease course. The formation of these proteins is similar to putting together your own furniture, let's say an IKEA chair. Your instruction manual informs the formation of the chair, much like RNA informs the creation of protein inside a cell. Now, you're eager and you want to work quickly, so you don't follow the instructions very closely, instead just skipping to the middle of the manual or leaving a few steps off at the end. What you get is something that looks like a chair, but its haphazard construction creates an unintended product. And in the case of the brain and spinal cord, these proteins or your furniture can have toxic consequences. They can take up space in the cell they shouldn't and interfere with the cell's normal functions, creating an environment that probably doesn't spark joy. Now, a lot of people like to blame the instruction manual, but what if ALS could be treated simply by cleaning house? The goal of my thesis is to understand how these unexpected proteins affect the overall health of motor neurons and muscle. I create models where I can look at these proteins without other confounding factors and see what changes occur as they would in ALS, such as cell death or movement deficiencies. Our lab has been able to demonstrate that these proteins can be toxic to isolated cells. So the next step is to see what happens to networks of cells as well as animals. Not only will these models help in our understanding of what these proteins do, but they'll also help in screening potential therapies to treat this ALS. And if treating this ALS is as simple as tidying up and eliminating the toxic proteins, maybe we can help change a few of the 15 diagnosed ALS patients each day into ALS survivors. Thank you.
Thank you, Brandie.